Welcome to Grovebio channel. In today's video, we will be dealing with DNA replication of prokaryotes. In this video, we will be dealing with termination. In order to know initiation and elongation, you can either click in the description below or the icon above. Now we can see about the termination. Termination. The gene and enzyme involved are TER sites. This is gene site. There are nearly 7 sites. TER A, B, C, D, E, F and G. And we are in need of very important protein named TUS protein, TUS protein. And along with it, we need specialized topoisomerase and with the presence of part3 and part3 gene. These genes will be responsible for separating the daughter strands so that 2 daughter DNA will be produced at the end of replication. Now we can see about the termination. The termination of prokaryotic replication done by its terminator site, that is, it has non-palindromic 23 base pair. It means it has no repetitive sequence. So here you can see the covalently closed circular complex with ORIC present. So this looks like theta. So it is known as theta mode of replication. So here you can see TER EDA and TER GFBC at the termination site. The TER EDA, these 3 sites stops the counterclockwise traveling of the fork. That is, as you can see, the newly synthesized strand of the blue strand travels in the anti-clockwise direction. As we saw in the initiation video, as I said, it is very important that one strand will be traveling in anti-clockwise direction and other strand in clockwise direction. So in termination, it is very important. So the newly synthesized strand from the blue template will be traveling in the anti-clockwise direction. So when it encounters the TER EDA, these 3 sites at the blue strand, what happens? It sees the replication. Similarly, the newly synthesized strand from green strand, it travels in the clockwise direction. When it encounters GFBC site, TER sites at the green strand, what happens? It stops the further replication. The TER sites arrest the motion by using TUS protein, the very important protein, so that it will arrest the motion of the replication fork. It prevents strand displacement by the DNA B helicase so that further unwinding of DNA will be prevented, so that DNA helicase cannot be moved. And after that, topoisomerase along with part C and E genes, these 2 genes along with specialized topoisomerase separates the strand. So at the end of replication, 2 daughter DNA will be produced, thus the termination. Now we can see about summary of DNA replication in prokaryotes. At first, ORIC region, that is chromosomal replication initiation region, it has 9 mers and 13 mers. At the 9 mers, the DNA A will be binded. 6 to 12 DNA A binds to it, and each DNA A will use ATP and forms the replication bubble at 13 mer by melting the DNA. So that in 13 mer, DNA melting occurs, and after which the helicase binds to the replication fork so that it unwinds the DNA. So the helicase is also called as DNA B, and it unwinds the DNA and forms the replication bubble. And after which the SSB protein, the active form is tetramer. The tetramer SSB protein binds to the single-stranded template DNA to prevent the base pairing between the templates, that is blue and green templates. And after which, when the helicase moves, it causes supercoiling. In order to avoid that, topoisomerase 2, it is also called as gyrase, it binds to the DNA and it avoids the positive supercoiling caused by the helicase. How it works? It either breaks the DNA or it causes negative supercoiling, so that when the positive supercoiling occurs, it undoes its effects. And next is RNA primase. The work of RNA primase is to add the first nucleotide without the 3' OH. So the RNA primase adds the RNA primer in both leading and lagging strand. So one primer will be at the leading strand and more primers will be at lagging strand. And after which DNA polymerase 3 acts the nucleotides by extending from the 3' OH, which is created by the RNA primase. So in both leading and lagging strand, it extends the nucleotides. And after which the DNA polymerase 1 along with the RNAH removes the RNA primer and DNA polymerase 1 fills the gap created by the RNA primers. So RNase H removes the RNA primers and DNA polymerase 1 adds the DNA primer in it. So it has special capability at 5' to 3' exonuclease capability and 3' to 5' polymerase activity, so that it adds the polymer at this end 3' to 5' end. The DNA polymerase 1 can work without the ATP. It also has the special capability C 3' to 5'. Other DNA polymerase can only add nucleotides along the 5' to 3' direction of the newly synthesized strand, whereas it can add nucleotides in the opposite ways 3' to 5', so that it won't disturb the helicase activity. And next is ligase. The DNA ligase seals the nick, that is small space between the nucleotides, and after which the TER site, when encounters along with TUS protein, it seals the replication, and then topoisomerase along with part3 genes separates the DNA strands so that it forms two-daughter DNA. Do you know, in DBT BED 2008 question, which of the following not found in E.COLI replication fork? Options are DNA A, primase, pCNA, SSB. pCNA is like beta clamp. SSB protein is also present. Primase, RNA primase is the starting of elongation. And DNA A is present in initiation. The question is not found in replication fork. The DNA A is not found in replication fork because it only creates the melting of 13-mer, but it is present only in the 9-mer. It is not present in the replication fork. So the answer is DNA A. Thank you for listening. If you have any doubt, comment in the comment section below, subscribe and please press the bell button below. Thank you so much for listening.